Hello everyone and welcome. In this video, I'm going to talk about FDA's umbrella pathway for authorization of antibody tests during the current COVID-19 crisis. One test has already been authorized under this pathway, and I've talked about that in a video. You can find that video on my YouTube channel. But in this video, I'm going to go into a little bit more detail about the requirements and the kind of samples that FDA is using during the independent validation part of the authorization. It might give you insights if you are trying to develop your own test and thinking about how to design a validation study. It'll give you some helpful hints. So let's look into that. What is the umbrella pathway? FDA came out with this authorization pathway in late April, and it applies to antibody tests using lateral flow or ELISA techniques. But the key requirement, as highlighted here, is an independent validation study performed at the NCI or another agency designated by the FDA. What are the performance requirements? So let's look into the expectations for positive agreement and negative agreement. FDA will conduct those tests. Sample type will be serum and plasma. 30 confirmed positive samples will be tested, and overall expectation for positive agreement is at 90%. Individually for IgM at 70% and IgG 90%. Negative agreement, again, established using serum plasma samples. 80 confirmed negative samples or pre-COVID-19 samples will be tested. 10 of them will be HIV positive. And the overall expectation is at 95% negative agreement and no cross-reactivity with HIV. Now, you may have heard terms like diagnostic sensitivity and specificity. It's the same thing here. Positive agreement is diagnostic sensitivity. Negative agreement is diagnostic specificity. So it's the same thing. To me, most interesting part is the composition of the panel FDA is using for validation of these tests. And this comes out of a report that was just published, and it's on FDA's website for the first test that got authorized under this pathway. These 30 samples contain both IgM and IgG at different titer values. For IgM, most of them are below 1600, but for IgG, it's a broader range. However, about 50% are at about 1600 titer level. So it covers a broader range and provides a whole distribution to be tested and validated. Another interesting thing is to see the distribution of number of days from onset of symptoms reflected by these samples, and it ranges from 17 to 36. However, most of these samples are collected within 17 to 24 days. So this is a key point. The performance of these tests validated this way is only during this time frame, and antibody tests as a result may not be very sensitive if they are performed very early on during the infection. And that could be one of the problems in the field, and maybe people are not finding these tests to be as accurate as they would like it to be. So something to carefully think about when to do this type of testing. For negative samples, as I mentioned before, there'll be a total of 80 samples prior to 2020. That's what they're using. Clinical status, 70 samples without regard to any clinical status. But notice they have included 2 samples that showed cross-reactivity in other tests. 10 samples will be with HIV positive and 3 samples have shown cross-reactivity in other tests. So what FDA wants to also establish that there is no cross-reactivity to these negative samples. That's a good thing for a test manufacturer, because if you are able to validate performance this way, you don't need to do a whole lot of cross-reactivity studies. One exception is class specificity. You still have to conduct that. Now, in addition to these performance requirements, you can conduct additional studies and produce that information to the FDA, but this is the bare minimum. There are a lot of labeling requirements that you have to be mindful of, and they are mentioned in FDA's letter of authorization. What is the intended use? Does it detect IgM, IgG, or total? You have to specifically mention that it's supposed to be used only under this EUA, Emergency Use Authorization. It's not intended to diagnose acute SARS-CoV-2 infection. It is not a diagnostic device for the infection. What it tells us is the immune response to the infection. You have to mention that negative results do not preclude infection, and false positive may occur due to cross-reactivity. So these disclosures have to be made in your labeling. You have to provide detailed instructions for performing the test and interpreting the test results. This is by far the longest section in the IFU. And date indicating when the IFU was generated. You have to detail the study descriptions and results in addition to this independent validation done on the test from all other performance testing you might have conducted. You have to mention the limitations, for example, deviations. What are the conditions of testing under which the test may not perform very well? And you have to say that it cannot be used as a sole basis of patient management decisions. You have to provide that limitation. And you have to provide manufacturer contact information. So there's a lot of labeling requirements. I would recommend reading the EUA carefully to make sure you are covering all of those requirements. FDA is going to publish all the tests authorized under this program in Appendix A, and that will be on FDA's Emergency Use Authorizations website. You can find that on Google. So far, only one test has been authorized, as I mentioned before. It's from Euroimmun US, and it uses the ELISA technique. It is a fairly good test, and I have covered its performance requirements in another video that you can find on my YouTube channel. So this is very good news because it offers a right mix of validation done independently. It reduces the testing burden on a manufacturer, and it offers an alternative to the EUA pathway. But bear in mind that it is getting much more difficult now because people know about it already. And in one of the FDA town halls that I attended recently, it was mentioned that there is now a backlog of testing at NCI. So you may have to wait. And there might be a trade-off decision here, how quickly you want to get in versus how much testing you want to do it on your own. So you have to consider those and make those decisions carefully based on your timeline. But I hope this is helpful. A lot is happening. Things are happening very fast. FDA is coming out with many EUAs and many different tests are coming in the market. So a lot to catch up. The best way to keep in touch is to subscribe to my YouTube channel, and I'll show you how to do that. You can go to my YouTube channel and subscribe, or you can follow me on LinkedIn. Whenever I post anything on LinkedIn, a video like this, I will give you a link to come back to my YouTube channel, look up my other videos and subscribe. I would like to hear your questions and comments. What are you concerned about? What are you thinking that still remains unanswered? And I'll be very happy to produce another video for you. Finally, I want to thank you for your interest and attention, and I hope all of you are staying safe.